Hello everyone, welcome to today's session. Today we are going to see class 12 biology, chapter 5, molecular basis of inheritance. In that we are going to see DNA fingerprinting. What is DNA fingerprinting? So DNA fingerprinting is a technique. It is used to identify and compare individuals based on differences in the DNA. So we have seen in our previous videos that 99.9% of the human DNA, those sequence is same among all human beings. But the remaining 0.1%, right, it shows variations between the individuals. And these variations, they are responsible for making each individual genetically unique. And if we consider the human genome to contain approximately, we already know, no, 3 into 10 power 9 base pairs, then the difference can involve approximately 3 into 10 power 6 base pairs. Now, sequencing the entire DNA of two individuals every time we want to find their genetic differences, it will be very difficult and also is time consuming and expensive process. So, in order to answer these, DNA fingerprinting, it provides a quick method to compare the DNA of two individuals. Yes. So let us just think of a human DNA as a very large book. So there, the most pages are almost identical in everyone. Only certain pages they contain the differences, and the DNA fingerprinting it focus on these highly variable regions instead of comparing the entire DNA sequence. So next we are going to see the repetitive DNA. So DNA fingerprinting, it identifies the differences in specific regions of DNA called the repetitive DNA. So repetitive DNA, it consists of small stretch or a short stretch of DNA that is repeated many times, that are repeated many times. For example, if we let us take this ATGC. So ATGC, ATGC, ATGC. So what happens here? The sequence ATGC is repeated several times. And when DNA is separated using this density gradient centrifugation, you can able to see here, yes, this diagrammatic part. So it is separated using the density gradient centrifugation. The bulk genomic DNA, it forms a major peak here. And the repetitive DNA, it forms additional smaller peaks, and these are called as the satellite DNA. And how they are, on what basis they are separated. See, based on the factors such as like a base composition, length of the DNA segment, and number of repetitive units. And again, the satellite DNA, it can be differentiated into two. That is, one is the microsatellite and another one is the mini satellite. And these sequences, they do not code for any protein, but they show a high degree of polymorphism. So this high degree of polymorphism is the basis of the DNA fingerprinting. Yes. So next we are going to see polymorphism, the basis. So what is this polymorphism? See, polymorphism means variation at the genetic level. Polymorphism means variation at the genetic level. So this arises due to mutation. Yes, and how, where this occurs? See, mutation, it can occur in a somatic cell, that is nothing but our body cells, or germ cells, that is which produces the gametes. So if a mutation occurs in a germ cell and it does not seriously affect the individual's ability to reproduce. So it is not going to affect the individual's ability to reproduce. And that mutation, it can be passed to the next generation. So what happens? So over many generations, such inherited variation, they may become common in a population. And this DNA polymorphism is described when more than one variant, or allele exist at a particular locus in a population. So with a frequency greater than 0.01. So how we can say? So when more than one variant or allele exist at a particular locus in a population with a frequency greater than 0.01. Or in a simple word, we can say this DNA polymorphism is an inherited variation in DNA that occurs at a significant frequency in a population. So this non-coding, yes, the non-coding DNA generally it shows greater polymorphism. Why? See, it shows greater polymorphism because mutation in these regions may not have an immediate effect on reproductive ability. So it is just a pass over the generation, right? So here you can able to see the diagram. So one person, one pattern they have taken. So let us take this the blood, the skin and saliva. Yes. So it shows one individual produces the same DNA fingerprint and the same principle applies to other tissues also here the hair, bone and sperm also. So because the DNA fingerprint is inherited, it can also be used in paternity testing. So next we are going to see the technique, VNTR and steps. So the technique of DNA fingerprinting, it was initially developed by Alec Jeffrey. Yes, it was developed by Alec Jeffrey. So he used a highly polymorphic satellite DNA sequence as a probe. Yes. So he used a highly polymorphic satellite DNA sequence as a probe and called that as the VNTR. And what this VNTR? VNTR is nothing but the variable number of tandem repeats. Variable number of tandem repeats. And this belongs to the mini satellite class of satellite DNA. It belongs to the mini satellite class of satellite DNA. So here a small segment, a small DNA sequence is repeated many times in a tandem arrangement. What happens here? A small DNA sequence is repeated many times in a tandem arrangement. And here the most important point is the number of repeats varies from one individual to another. For example, let us take three people A, B and C. And 1, 2, 3 will be the numbers. So in person A, it is 1, 2, 3 means, and the person B, it will be 1, 2, 3, 4. In the person C, it can be 1, 2 or 1, 2, 3, 4, 5, 6. Yes. So because the number of repeats is different, the length of the VNTR region also differs between the individuals. And this VNTR, they show high degree of polymorphism. It shows a high degree of polymorphism. Next is this the classic method. So the classic DNA fingerprinting technique, it uses the southern blot hybridization and it uses the radio labeled VNTR probe. So if I tell like that, you cannot understand. I'll teach you the steps also easy for you to understand. We'll see it. So each step I will explain. So steps of the technique. So the first step is isolation of the DNA. So DNA is first isolated from the biological sample. What kind of biological sample? See, the DNA can be obtained from the blood, hair follicle, saliva and other tissues also. Next is the digestion with the restriction enzymes. So the isolated DNA is now cut into fragments using restriction endonucleases. So the what this enzyme does, these enzymes, they cut the DNA at specific recognition sites. Only they cut the DNA at specific recognition sites. And next is separation by electrophoresis. So the DNA fragments, now we separated, right? They are separated according to their size using gel electrophoresis. So we have the fragments now. So the DNA fragments are separated according to their size. So size wise we are going to have the DNA fragments. How it is done? By gel electrophoresis. So the smaller fragments, they move faster and farther than the larger ones. And next technique is the blotting technique. So in blotting, the separated DNA fragments, they are transferred from the gel onto a synthetic membrane. So from the gel, yes, they are transferred to the synthetic membrane. So the membrane may be made from nitrocellulose or nylon. And this transfer is called as the southern blotting. Yes, nitrocellulose or it can be nylon, and this transfer is called as the southern blotting. Next technique is the hybridization. So the DNA fragments on the membrane, they are treated with a labeled VNTR probe. So they are treated with the label VNTR probe. The probe binds to its complementary DNA sequence. What happens here? We have given the probe over here, that is a VNTR. It goes and binds to its complementary DNA sequence, and this process is called as hybridization. And next process is detection by autoradiography. So the hybridized DNA fragments, they uh, are detected by autoradiography. What happens? Already we know the VNTR probe. Yes, it goes and binds with the complementary strands. Now it can be detected by autoradiography. So this produces a pattern of bands, and these bands together forms the individual characteristic DNA fingerprint. Yes, you can able to understand all these things stepwise. You can ask questions in the comment box, children. Yeah. Uh, yes, you can ask. I'll answer to you immediately. Reading a DNA fingerprint. So after the DNA fingerprinting process, the autoradiograph produces many bands of different sizes, And why they are producing different sizes, bands of different sizes? Because they are nothing but the characteristics of an individual. And the number of VNTR repeats varies between individual. The DNA fragments produced also, they differ in size. That is why the banding pattern is generally different for every individual. Yes, it is unique per person. Yeah, it differs for every individual. But here one exception is there. That is the monozygotic or the identical twins. They have essentially the same DNA. And here is some diagram for you. A crime scene DNA has been matched against individual A and individual B. So the banding pattern, the DNA banding pattern obtained from the crime scene is compared with the patterns of A and B. Yes. So what it demonstrates? See, the figure demonstrates that the crime scene DNA matches individual B and not with the individual A. You can able to see here red band, here red band, here. Yes, yes. So which one is matching B? Yes. So just think. Yes, these barcodes you can easily able to find the person. So next we are going to see. Let's have a quick revision. DNA fingerprinting means it's nothing but a quick method to compare the DNA and repetitive DNA. They are short DNA sequences repeated many times, and satellite DNA, they are repetitive DNA appearing as minor peaks, and polymorphism is inheritable variation at the genetic level, and VNTR is a variable number of tandem repeats and is a mini satellite probe. And who is Alec Jeffrey? He developed the DNA fingerprinting technique. And sudden blotting is a transfer of DNA fragments to a membrane. What is this PCR? PCR is nothing but polymerized chain reaction. So it increases the sensitivity, and also DNA from a single cell can be sufficient. And some applications, these things are used in forensic science, paternity testing, and also population and genetic diversity studies. So we have come to the end part. Is go through whole video, children. Don't miss it. We'll see you in the next video. Thank you.